My name is Eric Johnson. I am the field application scientist for the Southeast region for Bio-Rad. And welcome everybody to the second seminar focusing on Droplet Digital PCR, covering how our customers are using Bio-Rad genomics products in the fight against COVID-19. So just a quick agenda for today's presentation. I'm going to very briefly introduce what is Droplet Digital PCR for those of you that were on the call last week. A lot of the same information, but I am anticipating that we're going to have some new people on the call today. So we're going to very quickly go through an introduction into what is ddPCR, a very quick overview of data analysis to set up our applications, primarily focusing on COVID-19 research. Just a few housekeeping things. Everybody was muted upon entry. If you can, please keep yourself muted throughout the presentation, just to keep background noise to a minimum. I urge everybody to ask questions as we go through this presentation. You can either ask the question in the chat box, and I will try to answer those as we go along. I will also stop at different points during the presentation if you would like to unmute yourself at that time and ask a question. I will also, just like last week, be sending out a PDF version of these slides. So if you want these slides, you will have these, as well as some additional information. And most importantly, Lisa, your genomic sales specialist, is also on this call. If you guys are interested in looking into obtaining or running Droplet Digital PCR, I would urge you to either message her in the chat, or you can email her. I will be providing her email address at the end of the presentation. So she is your sales specialist. She's probably, in some sense, the most important person on this call. So let's go ahead and get started. So for those of you that were on last week, you'll remember that the major difference between traditional and qPCR approaches versus our new evolution of Droplet Digital PCR is the way that we process the sample. So with traditional or qPCR, we have one bulk 20 microliter reaction that gets thermocycled to endpoint, and then we are either loading that on a gel or monitoring changes in fluorescence with real-time PCR, generating amplification plots and getting a CQ value. Now with Droplet Digital PCR, we're going to start with the same bulk 20 microliter reaction, but we are going to first dropletize this and turn it into 20,000 1 nanoliter size droplets. We are then going to be counting droplets one at a time and looking to determine whether or not a droplet contained our target of interest, yes or no. If it does contain our target, that is going to be positive for a molecule of interest and a fluorescent droplet. If it does not, it is no fluorescence, negative droplet. Okay. So the defining difference between your traditional and qPCR approaches versus ddPCR is just the conversion of the reaction to droplets. Now, at Bio-Rad, we like to call this the power of partitioning. So if we were to take a theoretical example of a 20 microliter reaction that contains 6 molecules of our target, let's say 6 molecules of the virus that causes COVID-19, and we go to dropletize that into 20,000 1 nanoliter size droplets, by random distribution, we are expecting that we are going to get about 6 positive droplets. In this case, the droplets that are shown here in green. Now, each PCR or each droplet represents an independent single amplification event. So if it contains a molecule of interest, PCR is going to occur inside that droplet, and we are going to generate fluorescence. So that is going to be a highly fluorescent positive droplet, which we are going to call a 1. Now, if it does not contain a molecule of interest, PCR is not going to occur. Therefore, we are not generating any fluorescence, and that is going to be a negative droplet that we're going to call a 0. So the data is effectively compressed down to 1s and 0s. And that is where that digital name in Droplet Digital PCR comes from. Now, the number of positive and negative droplets is going to be directly proportional to the initial starting concentration of the sample. So if we were to go from a sample that has 0 molecules of our target DNA, we are going to get 0 positive droplets. Now, as we start to increase the concentration of our target molecule of interest in sample 1, 2, and 3, going from a low, medium, and high concentration of our target, you will see that we get more and more positive droplets. So effectively, all that we're doing here is counting droplets. ddPCR is just a very fine-tuned droplet counting machine. Now, the ratio of positive to negative droplets, or using the ratio of positive to negative droplets and utilizing Poisson statistics, we're able to calculate an absolute concentration in copies per microliter without having to run a standard curve. So every sample generates an absolute quantification. Now, one question I do get asked on here, and we're going to talk about multiple occupancy later on, is can you have multiple copies of the same target or multiple targets in the same droplet? And yes, you can. As the concentration of our target increases, so does the likelihood that through random distribution, multiple targets go into the same droplet. However, Poisson statistics accounts for that during the calculation. So even though we do have multiple occupancy, that is accounted for with Poisson statistics and is accounted for in your absolute concentration measurement that you get after running ddPCR. Okay. Is there any questions? I know that I went through that rather quickly. I want to make sure that we have enough time to talk about the applications later on. Is there any questions about the theory behind ddPCR? Okay, so let's quickly go through what does the workflow look like. So for a reaction setup, you're going to set up your samples just like you do with traditional or qPCR. It uses a 2x super mix that has TAC, DNTPs, and magnesium chloride and buffers, etc. You're going to add your specific primer and probe, your cDNA, RNA, or genomic DNA template, and then water to bring it up to a 22 microliter reaction. Okay. And it is compatible. We have super mixes for both looking at probe chemistry, so FAM and hex probe, or dye-based chemistry, which would be equivalent to cyber green by qPCR. We also even have a super mix that is a one-step super mix that you can take an input of RNA. It'll do reverse transcription, so it'll convert your RNA into cDNA and then do the amplification all in droplets. And that's the most common super mix that we're seeing groups using for COVID-19 research because it is an RNA virus. Now, once we set up our reaction, we then have the added step of generating our droplets using our droplet generation cartridge. This cartridge has built-in microfluidics. It's going to combine sample, which is coming in left to right, oil, which is coming in from the top and the bottom, to generate droplets that are water-oil immersions that are about 1 nanoliter in volume. And it'll process 8 samples in 3 minutes, a little more than 30 minutes to do a full 96 well plate, which is 96 individual samples. So very quick. Once we've generated our droplets, we are going to then thermal cycle those droplets to endpoint. So this is our PCR amplification, is endpoint amplification. And then after we've amplified our target within our droplets, we are going to read those on our QX200 droplet reader and measure the level of fluorescence in droplets one at a time. So this droplet reader is going to pull up the droplets from each individual well. It's going to singulate them past a fluorescent detector, and then we're going to measure the level of fluorescence in either 1 or 2 channels. And then all of that data is getting displayed in our Quantisoft software, and we are able to very quickly get a concentration, an absolute concentration in copies per microliter, and you can see that down here. Okay, so how do we actually analyze data? So we're going to just very quickly go through data analysis and to set this up for talking about COVID-19. So if we start very simple with a singleplex assay, we can use either probe-based or evergreen-based chemistry. And we're expecting in this case, because we're looking at a single target, to get 2 populations of droplets that are either positive for FAM, or droplets where PCR occurred, so positive for FAM or positive for evergreen, and then droplets that are negative for fluorescence, droplets that did not contain our molecule. Now, we will visualize this on a simple 1D plot where you can see that fluorescence amplitude is on the y-axis. So the higher up the y-axis, the brighter fluorescent the droplet. And then on the x-axis is simply droplet number. And you can see in this case, I have my highly fluorescent positive droplets colored here in blue, my low fluorescent negative droplets down here in black. And then all we do is we set our threshold line to say any droplet above this we call positive for our target, any droplet below is negative, and then using Poisson statistics, we take the ratio of the positive and the negative and calculate absolute concentration. Now, for a duplex assay, we are going to be adding, or oftentimes this is done using probes, but we're going to add a separate primer and probe target. So for target 1, this would be with a FAM probe. Target 2 would be with a hex probe. Common applications for this include stuff like gene expression analysis, copy number variation, gene editing, as well as viral and pathogen detection, which we will be talking about for today's presentation. Now, in this case, we are expecting to get 4 populations of droplets. Now, because these are 2 independent targets, being target 1 and target 2 with independent primer and probe sets, we can analyze these by looking at individual 1D plots. So we can look at our 1D plot for FAM and our 1D plot for hex, draw our threshold line, and calculate our absolute concentration. Or we can look at this using a 2D plot where FAM is on the y-axis, hex is on the x-axis, and then we have our 4 populations, being our negative population, our droplets that are positive for our FAM target only, droplets that are positive for our hex target only, and the droplets that by random distribution have both targets and therefore are dual positive. And then all we have to do is set our threshold lines and we calculate an absolute concentration for both targets. Okay. Now, before we move on to talking about COVID-19, is there any questions as far as just general background when it comes to ddPCR? Okay. I'm going to take that as a good sign that I did such a good job last week that there's no questions. All right, so let's talk now about, in this latter half of this presentation, specifically about COVID-19, as well as the virus SARS-CoV-2, which is a virus that causes COVID-19. So shown here is a schematic representation of both the virus as well as the viral genome. So looking at the virus, you see it is an enveloped virus with on the inside is your genomic RNA. And then there are several proteins that comprise this virus, including the spike protein, which is S here. E stands for an envelope small membrane protein. M stands for a membrane protein, an additional membrane protein. N stands for the nuclear protein. Which binds, which are these little circles that are binding the genomic RNA. Now, the genome of SARS-CoV-2 is shown here. It is actually quite a fascinating and complex genome. It's a little more than 30 kb, and it's quite complex. It has multiple open reading frames, as well as several of these TRSs, which are transcriptional regulatory sites, and codes for a handful of genes. Now, when trying to detect the presence of viral RNA, either in clinical samples or in environmental samples, there have been several assays that have been generated for against this genome. The most common that we've seen are assays designed against and when I say assays, I just mean primer sets designed against the open reading frames. We've also seen several that have been generated against the E gene, and then most commonly are assays that have been designed against the N gene, for which there are 2 common assays that groups are using, being the N1 and the N2 region of the N gene. Importantly, the N1 and the N2 were actually designed by the US CDC and are probably the most 2 common assays that groups are using for the detection of SARS-CoV-2 in a sample. Now, as far as Bio-Rad solutions that groups are using, we have kind of 4 main categories, being diagnostic and conformational testing. So this is, those qPCR tests where you go and you get your nasopharyngeal swab, and then they test that for the presence of the virus to confirm whether or not you are infected with COVID-19. We also have solutions for surveillance, whether that is surface testing, wastewater testing, or even air testing. We're going to be talking specifically about wastewater testing for today. And then there also have other solutions, including antibody detection, so serology testing to determine whether or not you are producing antibody or somebody is producing antibodies to COVID-19, as well as we are supporting vaccine and therapeutic research and development. Now, for today's presentation, we are going to be focusing kind of on these left 2 applications, being diagnostic confirmation and COVID-19 surveillance, as these really are focused on looking at RNA or viral RNA, and this is obviously a talk about or this is a genomics talk. So first, let's start by talking about diagnostic and conformational testing. So there are Bio-Rad is supporting both qPCR and ddPCR approaches for SARS-CoV-2 detection for those clinical diagnostic and conformational testing. For qPCR, although we have not produced our own kit, there are several kits that are out there that have gained emergency use authorization, or EUA for short, approval from the FDA that utilize Bio-Rad CFX real-time instruments. So we are supporting it on the instrument side. This includes assays that have been designed to be real-time assays, as well as LAMP assays, so thermal isol amplification, as well as kits that use either extraction methods such as column-based or bead-based extraction, as well as a couple of kits that are using extraction-free methods. So we are supporting qPCR on the instrument side. Now, for the Droplet Digital side, we have developed a kit for SARS-CoV-2 detection that also has gained FDA EUA approval. This utilizes our QX200 AutoDG platforms, including our research use only platform, as well as our in vitro diagnostic, which is a clinically our clinical instrument that has FDA approval. As far as sample types, we're going to use it can handle nasopharyngeal swabs, as well as wash, as well as a couple of other ways of collecting the material using swabs. And importantly, and this is going to start deviating from what we've talked about up to this point, is that this kit actually uses what is called a triplex assay. So this kit looks at both the N1 and the N2 target, so those assays that we talked about, as well as an additional target being the RNase P gene, or RP for short. And we analyze all 3 of these targets in the same well. So up to this point, we've talked about singleplex and duplex assays in the same well. We are now adding an additional target and looking at 3 targets all in the single reaction. So how do we go about looking at more than 2 targets when we only have 2 channels for fluorescence? Well, we call this advanced probe multiplexing. And we've got this for both triplex assays, looking at 3 targets, as well as there are examples of groups doing 4plex. And I've seen an example of a group that's even looked at 5 targets all in the single well. And we do this through some kind of unique, cool modifications of the reaction setup. So let's talk about how we're able to do advanced triplex multiplexing. So in this case, we've got our N1, our N2, and our RP gene, so target 1, 2, and 3. Now for target 1, which is N1, this is going to be 100% FAM. Okay, so the only probe in there is a FAM probe. Now, and that's going to be amplifying, for FAM fluorescence. Our target 3, which is RP, is going to be is 100% hex. So if you think back to the duplex assay that I showed, N1 and RP, or target 1 and 3, would be set up just like a standard duplex assay. Where we add to this is target 2, which is going to be our N2 target. And in this case, what we're doing is we are going to be using a dual probe, or two probes that are identical probes, but they're labeled with FAM and hex respectively. And each of these are going to be run at 50% the concentration, as opposed to 50% of the concentration compared to the N1 and the RP respectively. So if we look down here at our N gene, we've got our N1, which is 100% FAM. So anytime we amplify our N1 target, we are hydrolyzing just the FAM probe. Now for N2, again, it is an identical probe. The only difference between this probe is that one is labeled with FAM and one is labeled with hex. So if we think about what's going on inside of a droplet when we have our N2 target, 50% of the time that we amplify the N2 target, we hydrolyze a FAM probe, and the other 50% we hydrolyze a hex probe. So at the end of 40 rounds of amplification, our droplets are going to be dual positive for both FAM and hex. Now, because we are running these at a 50% concentration, the overall fluorescence of FAM and hex is going to be lower than if we were to run it with 100% FAM or 100% hex. So if we look at kind of a cartoon representation of where these droplets are going to be, that is shown here. So let's just kind of go through this so everybody's clear on how we're doing this triplex assay. So down here in gray is our negative droplets, right? That are negative for all of our fluorescence. If we look at our N1, which is 100% FAM, but no hex. So again, on our y-axis is FAM fluorescence, on the x-axis is hex fluorescence. You can see that we have a population straight up from the negative cluster that only has FAM fluorescence. So this is our N1. Additionally, for RP, which is 100% hex, it only has hex fluorescence, so it's only moving on the x-axis, and that's shown here in purple. Now for N2, because every time we amplify N2, we hydrolyze 50% FAM and 50% hex, this population is going to have dual fluorescence, but it's going to be at a lower fluorescence than if we were to run it with 100% for either FAM or hex. So if we go halfway between N1 and our negative, and then over halfway between our negative and our RP, we have our N2 target. All right, so it has half the fluorescence of FAM and half the fluorescence of hex, which slides it down in between. I like to think of this kind of like a baseball field. So you've got home plate, right field, center field, left field. Okay, now this is the easy part. Where it gets a little bit more complicated is when you start having multiple occupancy, which we absolutely can have if our concentrations get high enough. So the easiest one is if we have a droplet that contains N1 and RP, which is going to be 100% FAM, 100% hex. So we come up to our N1, over to our RP, and then here in light blue has the same fluorescence for N1 and the same fluorescence as N2 is our dual positive N1 plus RP. Now, what about the scenario where we have N1 and N2 together? So we've got 100% FAM plus 50% FAM. So we're going to go 100% FAM plus an additional amount of FAM, and then 50% FAM. So over to where N2 would be, and that is our N2 plus N1 population. Same thing for N2 plus RP. We've got 50, 100, 150% hex, 50% FAM, there's our N2 RP. And then in the upper right hand at 150% the fluorescence of FAM and hex is our triple positive droplets that have all three targets. So you can see here that all we're doing is adding fluorescence. It's additive. So 50 plus 150 gives me 150% fluorescence. Okay. And importantly, this, the example that I'm showing you here is for SARS-CoV-2. This can actually be done on any assay or any set of samples that you want to look at three targets. So it's not specific to SARS-CoV-2. It's just something that you can do with several targets. And there are several examples of triplex assays that are out there. Now, this is a cartoon representation. What does the actual data look like? So here is an example data set using our triplex assay. Again, you could see our negatives. We also have our N1, which are positive for FAM only, our N2 that are our RP that are positive for hex only, and then we've got our dual positive droplets that are positive for both, and then our 50% FAM and hex, which is our N2. So just like what we saw with the cartoon. And then, of course, we've got our N1 plus RP, which is just the fluorescence of N1 plus the fluorescence of RP. And then our 150% FAM, 50% hex, N1 plus N2, our N2 plus RP, and then our triplex assay, or our triple net positives that have the highest fluorescence for both FAM and hex. Okay, so even though we don't have that perfect blocking like we had here, all that I care about is I can distinguish these populations and I know about where to expect them. So then we can set our thresholding, and then the software can take all of those and calculate an absolute concentration for both the N1, the N2, and the RP. So very simple data analysis, as long as you can get separation of populations. Now, looking at the results looking at some results for some other examples where we're at very low concentrations, so down near the limit of the detection, you can see that in A, we only have 4 positive droplets, 2 droplets that are positive for N2 here, and 2 droplets that are positive for N1. However, because ddPCR is such a precise measurement, even with only having 4 droplets, we are able to identify with high accuracy and high confidence the presence of the virus in the sample. And then B would be another example that just has a slightly higher concentration, which is just giving us more positive droplets. But even in this case, we don't have RP, we don't have dual positives, and we don't have triple positive populations, but that is okay. All we have to do is identify one or the other. So if we look actually at the limit of detection of our assay, it's any sample that has greater than or equal to 0.1 copy per microliter, or in other words, 2 or more positive droplets. One can consider that a sample that is positive for SARS-CoV-2. CoV-2 has been detected. So if I go back, even this case up here where we only have 4 droplets, that's greater than equal than 2. So this is a true positive sample. So very high sensitivity with ddPCR for being able to detect this. And there are actually papers out there. We're not going to go over these papers today, but there are papers that have compared qPCR versus ddPCR on nasopharyngeal swabs and have identified that that ddPCR results in less false negatives than what qPCR has. So it's a more sensitive measure. Now, I'm not going to go over these bulletins. I just want to show you that Bio-Rad has recently come out with 2 bulletins using our triplex assay for both environmental swabs. So this would be surface testing, as well as nasopharyngeal swabs that use extraction-free processes. So typically, you go through a process of extracting the RNA. These protocols are highlighting an extraction-free method, which allows you to bypass having to go through extraction. And again, these can be applied to various other applications, whether they're other pathogens, E. coli, even single cells. Okay, so we've talked about Bio-Rad's approach, our Bio-Rad solutions for diagnostic and confirmational testing. In the latter half of this presentation, what I want to go over is COVID-19 surveillance, particularly focusing on wastewater analysis. So from last week's presentation, I showed this figure for the landscape of ddPCR application, which covered various applications that ddPCR is being used for. And I alluded to that for today's presentation, we were going to be focusing on looking at infectious disease and environmental monitoring. So for today, we're talking about COVID-19, which is an infectious disease, and we're looking at detecting that in environmental samples. Now, it's not, again, it's not, we're not specifically, or ddPCR is not specific for COVID-19 and wastewater. There are various applications for which we are seeing groups use ddPCR for, with samples that are notoriously difficult to work with because of the presence of PCR inhibitors. So this includes wastewater, or just surface water. I'm going to show you an example of looking at communal water, also soil samples, pretty much any type of bodily fluid, so blood, urine, feces, tears, saliva, as well as samples such as FFPE, which have fixatives, as well as a lot of chewed up DNA and high background. So all of the the common theme with all of these is that these are samples that have been notoriously difficult to work with by qPCR because of the presence of PCR inhibitors. Now, the reason that we're seeing groups transition to ddPCR for a lot of environmental testing is due to the fact that ddPCR has a lot of inherent flexibility built into it. And I want to talk about a few types of flexibility that we get with ddPCR. So the first is thermal flexibility. So when we're talking about thermal flexibility, what we're looking at is a range at which the assay can be run at. So in this case, in the example that I'm showing you here, we've done a thermal gradient of 8 technical replicates between A and H, going from 65 down to 55 degrees. Now, at 65 degrees, our highest temperature, you can see that the positive and the negative population are right on top of each other, right? We would have a hard time looking at this sample and setting a threshold to distinguish where do the negatives end and the positives begin. As we start to cool the reaction down, however, you can see the positive droplets are getting brighter and brighter, all the way up to about G or H, where we now have very highly fluorescent positive droplets. What is going on here is that as we cool the reaction down, the efficiency of the PCR reaction is increasing. Therefore, we are hydrolyzing more probe or binding more evergreen, and we're getting brighter and brighter droplets. However, looking at this plot, as long as I can distinguish a positive from a negative droplet and draw my threshold, I can use that temperature. So I would be very comfortable and confident comparing the results between D, E, F, G, and H, because I have a very clear separation between my positive and my negatives. The total number of positive droplets is not changing, just the level of fluorescence of those droplets. So the concentration that we calculate from D through H is going to be the same value. So a very large range at which we can use, a very large temperature range at which we can analyze with ddPCR, again, because it's endpoint measurement. Now, the next topic of flexibility is amplicon size. So in this example, what we're looking at here is varying amplicon sizes ranging from 400 to 5.7 kb amplicon. Now, typically when we're thinking qPCR, it's often recommended that you keep your amplicon between 50 and 150 base pairs. We don't want to go much over 150. The reason for that is that as you start getting past 150 base pairs, the efficiency of your amplification starts to decrease. So when I say efficiency, what I'm talking about here is that with qPCR, or with any type of PCR, every time you perform an amplification step, you, in theory, double the product. Now, if you exactly double it every single time, that is an efficiency of 100%. Now, if we're not getting 100% efficiency, let's say we're not doubling the product, our efficiency goes below 100%. Now, unfortunately, with qPCR, we are very tied to efficiency. We need to be operating around 100% efficiency. As we start to decrease our efficiency, if it gets low enough, the math and our confidence in our results become less reliable. So when we start doing these absolute quantification or these delta CQ comparisons, the math is not as robust when our efficiency is below, when it gets below 100%, typically below about 90%. So in this case, if we're looking at amplicons going from 400 up to 5.7 kb, so we're already more than double the size of what we can do by qPCR, you can see that we are getting positive droplets. However, there is a lot of these kind of intermediate droplets, often referred to as rain. And I would have a hard time setting my threshold in this case. But because we are doing endpoint amplification, that gives us flexibility when it comes to modifying the thermal cycling protocol. So in the standard two-step, where we're going between 95 and let's say 50 degrees, and we're doing this 40 times to amplify our product, we can instead design a three-step protocol where we do 95 degrees, then 55, then 72. So in some sense, this is your qPCR protocol, and this would be your traditional PCR protocol. All that we've done here is we've added more time for the enzyme to complete the amplicon. And what that does is allows us to push our amplicon size, in this case, all the way out to 2.5 kb, where we've increased the fluorescence and we've significantly reduced the rain. So now we can start looking at larger amplicons. This comes into play when you're looking at trying to identify small differences, let's say between species or even splice variants. Now, in addition to doing three-step protocols, we can also just do a standard two-step, but make this step longer. So as opposed to doing 1 minute of amplification, we can make it more like 2 or 3 minutes of amplification. The other thing we can do is also just add more cycles. So as opposed to doing 40 rounds of amplification, we can very easily go up to 45 or even 50 rounds of amplification, all with the thought process of we're just trying to boost the level of fluorescence in our droplets and separate our positive from our negative. Now, the last topic of flexibility that we're going to cover is inhibition and flexibility when it comes to the presence of inhibition. So I'm just going to show you one figure that highlights this. This is coming from Plant Methods Journal, where they are comparing qPCR in blue and ddPCR in green, looking at several different types of samples that are notorious for having PCR inhibition. And then they've on the y-axis is their percent inhibition as determined by measuring a sample with no inhibitors versus samples that have either high amounts, medium amounts, or low amounts of that inhibition. Now, what you can see pretty much across the board is that ddPCR is much more tolerant when it comes to PCR inhibition, right? Most of these lines are right around 0%, where you can see that qPCR has a lot of inhibition, particularly at high as well as medium, but also even at low amounts of the inhibition, including both wastewater, which we're going to be talking about in the next section, as well as humic acid, which is a component which is often found in wastewater as well. Okay, so in general, much more tolerant when it comes to inhibition. It's not that inhibition is being removed, the inhibition is still there, but because we're doing endpoint PCR, we're able to overcome that. Okay, so let's now talk about wastewater. And is there any questions before we go on to looking at wastewater? Any questions up to this point? Okay, so let's talk about wastewater applications. So wastewater has been a source of investigation for quite a while. And the reason for that is because our sewer systems accumulate samples from various individuals over a wide area, referred to as the sewer shed, and brings all of that sample to a central collection point. So as opposed to trying to measure samples from various individuals, you can get kind of, in essence, a pooled sample. It's because of this, and this is often referred to as wastewater-based epidemiology. And because of this, groups have been looking at various pathogens such as polio, influenza, HIV, also looking at the presence of E. coli, as well as even prescription drugs and biological weapons. It's for this reason that several groups have started to use ddPCR as well as qPCR to look for the detection of SARS-CoV-2 in the population. Now, there is several advantages when it comes to monitoring and looking to detect SARS-CoV-2 in wastewater. First, SARS-CoV-2 is shed in feces of both symptomatic and asymptomatic individuals. So even if you have a population that is not showing any symptoms, they are still spreading, or they are still shedding that into wastewater. So in some sense, it's kind of like an unbiased approach. Also, by monitoring for SARS-CoV-2, they can determine whether or not the virus is present or absent in the sewer shed. And then by doing absolute quantification, particularly with ddPCR, they can track trends over time. So are they seeing the virus increasing or decreasing based on its the viral load in wastewater? And then they can utilize that to project the trajectory of coming days of the virus. Do they see it going up, and do they need to prepare for that? Or is it going down, and maybe they can go into a new phase of reopening? Now, finally, there are groups that are working on determining if there's ways to estimate the actual prevalence in a community. We are not quite there yet, but there are groups that are certainly working on it, where they're trying to determine stuff like how many people are in the sewer shed, how often does somebody go to the bathroom, and can we, using that information, can we start determining the actual prevalence in the community? So putting a number such as a 1 in 1,000, 1 in 100,000 to that concentration, et cetera. Now, for those of you that are not familiar with wastewater testing, I certainly was not until COVID-19 came. I just wanted to just kind of quickly go over what it looks like or how these facilities operate. So you have your primary raw sewage inputs, which goes through primary treatment, including putting the raw sewage through screens to remove very large solid particles. It'll then go through a communitor. I'll let your imagination determine what that does. And then ultimately it goes through a primary clarifier. Now, out of this primary clarifier, you have two streams. You have primary effluent, which is your liquid, and then your primary sludge or raw sludge, also known as solids, which is going to be your solid material. Where the solid material is ultimately treated and disposed of, the primary effluent is also treated and then discharged as surface water. Now, as far as sample input for groups that are looking at SARS-CoV-2 in wastewater, they're looking at two primary sample inputs, being the primary effluent, being your liquids, as well as your primary solids, being sludge. Okay, now there are several, classically, ddPCR or classically qPCR has been used as the approach for studying wastewater. However, we're seeing a lot of groups starting to transition over to ddPCR because there are several challenges that are posed with qPCR that ddPCR is particularly poised to overcome. The first is qPCR is by nature semi-quantitative, and most groups are wanting to do absolute concentration. If you want to do absolute quantification by qPCR, it requires that you run a standard curve, which means you have to be able to generate a standard curve and maintain those standards, particularly if you're doing a longitudinal study. You need to make sure that you have those standards available throughout the entire study. ddPCR, however, is absolute quantification by nature, so there's no need to have a standard curve. The next challenge with qPCR is measurement variability. qPCR, oftentimes we're working down near the limit of detection when analyzing samples for wastewater. And anybody that has done enough qPCR where they're dealing with CQ values in the mid to upper 30s will know that those results are highly variable. Also, you can imagine that wastewater samples collected in the morning, the middle of the day, and the afternoon, as well as from plant to plant, are going to have various differences in the background and the amount of inhibition. Which also brings me to talking about low viral concentration for SARS-CoV-2. These groups are often working right down there around the limit of detection, where qPCR often struggles when dealing around the low limit of detection with variability and sensitivity. ddPCR, however, is extremely precise, and so very low variation between replicates, and is very accurate even down near the limit of detection. So very high precision and sensitivity compared to qPCR. And then finally, qPCR, again, is not tolerant when it comes to PCR inhibition. We need to make sure that we're getting rid of those inhibitors in order to get accurate quantification, where ddPCR, because it is endpoint PCR by nature, is tolerant to PCR inhibition. Okay, so in the last 10 minutes or so, I want to just quickly go through some examples of people using ddPCR and assessing water quality. So this first one is actually not looking at SARS-CoV-2. This is looking at fecal indicators in communal water. So I believe the primary source of their samples here is beach water. Highlights from this paper is they do a comparison between ddPCR and qPCR. They identify that ddPCR exhibits a higher precision and reproducibility, as well as more consistent results when looking at their targets, either in a duplex or simpleplex format, and that in general, ddPCR is about 1 or 2 orders more tolerant to inhibition than qPCR. So we're just going to look at one figure from this paper, which is shown here. So this group is looking at 2 targets, being Enterococcus, which is a general fecal indicator, as well as HF183, which is a human-specific fecal indicator, and they're looking to identify whether or not these indicators are present in communal water. So, is it present in the water near beaches? Now, they did look at, they ran qPCR and compared it to ddPCR. ddPCR was run either as a simplex, so each of these individually, or a duplex assay looking at both targets. qPCR was only run as a simplex because the duplex assay did not work by qPCR. And that's something that's not uncommon to see, that ddPCR is oftentimes a little bit easier to do multiplexing. But what you can see is that for both humic acid and natural organic material for both targets, qPCR in general was not tolerant to PCR inhibition. Even small amounts of the inhibitor resulted in large inhibition, where ddPCR was much more tolerant over a large range. Okay, the last 2 papers are specifically focused on COVID-19. The first one comes from California, and the goals here are looking to identify what is the best method for SARS-CoV-2 detection in wastewater, where they're comparing both qPCR and ddPCR, as well as some other comparisons such as extraction methods. Take-home message from this is they identified that solids, as opposed to liquids, are more sensitive for detecting the virus, and that viral load in solids correlates well with clinical confirmed cases in the sewer shed. So the first figure I'm showing you here is quite a complicated figure, but if we break it down, you can see that comparing influence versus primary solids, and this is all ddPCR data, that with ddPCR, the primary influence, only a subset of samples, so each of these are different dates of collection, only a subset of these were they able to detect either N1 or N2. And this was also consistent with qPCR. However, with primary solids by ddPCR, they were able to, for both N1 and N2, using 2 different types of extraction kits, they were able to consistently identify the presence of both N1 and N2 in all dates, with the exception of N1 on March 30th, and only the power fecal. So from this, they were able to conclude 2 things. 1, primary solids have about 100 to 1,000-fold higher viral concentration than influent samples because they are doing absolute quantification, and that they have different extraction kits, have different levels of inhibitors and different sensitivity when detecting SARS-CoV-2. Ultimately, though, they go on to conclude from this, and they didn't show qPCR data, but they go on to show that one-step ddPCR was much more reliable at detecting SARS-CoV-2 in primary solids than qPCR, and that is highlighted here. So not going to go through each one of these, but here are your individual dates comparing qPCR to ddPCR for solids, whether or not the RNA was isolated using a power fecal or RNA easy kit. But what you can see is that, and they did technical duplicates for both N1 and N2. But what you can see here is that in pretty much all cases, ddPCR was much more consistent at being able to identify the presence of N1 and N2, as well as being able to identify it in both technical replicates, where qPCR was much more sparse. So overall, more sensitive and reproducible with ddPCR than qPCR. Now, talking about qPCR, they did show, they did look at 2 controls, being an endogenous control, PPMOV, as well as an exogenous control, which is a bovine coronavirus that's being spiked into the samples, looking at undiluted, a 1 to 10, and a 1 to 50 dilution. And they identified that not only do they see varying inhibition by qPCR between two different facilities, facility A and facility B, but also from day to day, all the way up to about a 1 to 50-fold dilution, which, remember, we're working down near the limit of detection. At 1 to 50 dilution, they were not able to detect the virus by diluting it to the point that you were able to relieve the inhibition by qPCR. Now, looking at the last figure from this paper that we're going to look at, they compared confirmed clinical cases in the sewer shed versus the absolute concentration as determined by N1 and N2. And you could see that there is a very nice correlation between those. And they ultimately go on to conclude that collecting wastewater twice a week yields a significant association between the case counts as well as the viral load in the sewer shed. Okay, last paper that we're going to talk about, and then we're going to wrap up for today, is another paper looking at wastewater. This is the opposite coast, so this is in Virginia, specifically the Hampton Roads area of Virginia, again, where they're monitoring the presence of SARS-CoV-2 in wastewater. Now, the first figure I want to show is a very cool heat map that they generated going from March all the way to October. So this is a longitudinal study collected, and they collected wastewater at, I believe it was 9 different sites of collection. Now, and then they determined the absolute concentration as viral load over the course of several months in those areas. And what you can see here is that you can see kind of the ebb and the flow of the virus as it changes with the viral load in wastewater. One thing I do want to point out, which is very cool, at least to me, is if you keep an eye right on this little area right down here, in July, you're going to see a very prominent spike in the viral load in wastewater. So it's coming up here in just a second. So you see this very prominent spike that then decreases. This was actually associated with a cluster of positive cases on a naval base. And they were able to see that even in the wastewater. So I think a very cool approach. Now, looking at kind of a similar timeline going from March all the way to April, you can see that the viral load increases. They then enact stay-at-home order, and the virus decreases. And then as you go from phase 1, phase 2, and phase 3 reopening, you see this exponential rise in viral load as more and more people are getting out and interacting. Now, they've since updated this plot to include it going all the way out to October, as well as overlaying case count or clinical case counts for the people in that sewer shed. And what you can see is that as the case counts increase, there's a nice correlation of the rise of the virus. Now, continuing on for what I showed you on the previous slide, you can see that at the end of July, because of this increase in case counts, they actually had further Hampton Road restrictions. These are your classical restrictions that you think of, including limiting restaurant capacity to 50%, no large gatherings more than 50 people, et cetera. And that resulted in a decrease that was correlated with a decrease in case counts, as well as a dramatic drop in the amount of virus in the sewer shed. And then we'll see, as they've since gone back to phase 3 reopening, it'll be very interesting to see how this progresses moving forward. Now, just to wrap up, I do want to point out that there is a large collaboration of groups that are working on wastewater surveillance. I would point you to their website here if you are interested in wastewater surveillance. It is an excellent resource, so much so that even the CDC has started to jump on this as well. And recently, the CDC has generated a national wastewater surveillance system where they're working with towns, locales, cities, et cetera, for wastewater monitoring, where these groups can submit all of their data to the CDC for processing and generation of kind of nationwide monitoring of the viral load in wastewater. And I believe they're making funds available for this as well. So with that, we are right at time. I'm going to go ahead and wrap up. I will again send out a link to our system website that has a lot of great information, including our applications guide. It also will have a landing page for all of the COVID-19 applications that we talked about today. So with that, I will thank you all for your participation and listening to me talk, and wrap up by saying thank you. If you have questions moving forward, please do not hesitate to reach out to either Lisa or myself. So with that, thank you all, and have a great weekend. I will also stay on and ask any additional questions if there are any.